We're going to spend the last two lectures of this week, right before Thanksgiving break, on signal transduction. And what you'll see is that you learned a lot about signal transduction, but these lectures are really going to be focusing on the very end of signaling cascades, which is typically activating gene expression. So we're going to talk a lot about what lies at the end of signaling pathways and signal transduction pathways. And so today we're going to focus largely on transcription factors, and then on Friday we're going to talk a lot about how you assess transcriptional results in cells using modern methods, which is basically next generation DNA sequencing approaches. And incidentally, right after Thanksgiving break, I think it's on December 3, there is a lecture in the Department of Pharmaceutical Sciences seminar series, which is by Dr. Rachel O'Neill at MCB, and it's all about next-gen sequencing. So it kind of actually ties in with what we're talking about, and you can catch that. Okay, so as you have gathered, at the very end of signaling pathways, oftentimes what happens, you get a ligand binds to a receptor, second messenger systems are engaged, kinase cascades occur, but ultimately what happens in a lot of these pathways is that you activate the activity of transcription factors, and then those will go ahead and transcribe all the target genes that lie way downstream from a receptor. And that's what really makes the cell change and respond to the signal it received from a ligand. And so you've seen this before. Today, just to illustrate this aspect of signaling, we're going to talk about nuclear receptors. And these are transcription factors and receptors kind of combined into one, because they can bind the ligands, but then they get into the nucleus. In some cases, they reside in the nucleus all the time. But ultimately, that ligand binding causes them to associate with regulatory regions near the promoters of genes. And all the target genes for that transcription factor will get turned on, transcribed, made into messenger RNA, and then protein. So many proteins can be produced in the cell due to binding of these nuclear receptor type transcription factors. So first, a little bit generally about transcription factors. They work on DNA, obviously. And so here's a piece of DNA, and we're really focusing in on the regulatory parts of the DNA. So the gene is down here, but at the very beginning of the gene is a promoter, and Dr. Aniskevich talked about some of this. And of course, the promoter is where RNA polymerase can associate with that gene. And then once RNA polymerase is there, it will start to make a messenger RNA copy. But the interesting thing is that RNA polymerase on its own is not sufficient to perform transcription. It's actually subject to other regulation. And so what oftentimes happens, in fact, what usually happens, is that transcription factors first associate with the promoter region, and once the transcription factors have associated with the promoter region, then RNA polymerase is recruited and you get transcription going on. In addition to, so this whole region just upstream from the promoters where RNA polymerase and transcription factors associate. Another interesting DNA element is called an enhancer. And so enhancers are way upstream on the piece of DNA from the actual promoter. And it can be anything from 1,000 bases to even 20 kilobases upstream. So quite distant, really, from the promoter. And enhancers are specific DNA sequences, and activator proteins can bind enhancers, and then they help to recruit the transcription factors to the promoter. So whenever you have transcriptional activating proteins binding to an enhancer, typically they recruit all this other machinery, the transcription factors themselves, and ultimately RNA polymerase too, and then transcription can really get going. And oftentimes you'll notice that there's a big loop of DNA in between the enhancer and the promoter. So this site here all comes together in three-dimensional space to make a really potent transcription complex. And there are other things going on. So for instance, there are actually chromatin changes that can occur that regulate things, but we'll get into a little bit of that later. Okay, so obviously transcription factors are way downstream from receptors at the kind of the end of signaling cascades, pathways. And here's just one example of many. And it is an example using transcription factors called SMAD proteins. But what you can see up here is that what happens is ligands bind receptors, okay? And here are some of the ligands that bind these types of receptors. You know, activins, nodal, bone morphogenetic proteins, BMPs, all sorts of different pathways. The receptor is phosphorylated, but then the key is these SMAD proteins, which are actually transcription factors, they become phosphorylated, and then they can form trimers, okay? Dimers and trimers in some cases. And once these are formed as a multimer, they're now competent to be imported into the nucleus, and then they participate in turning on gene expression. And of course, many genes could have sequences that are similar that respond to SMADs. So this is typical of transcription factors. It's not just one transcription factor, one gene. Usually when one transcription factor is activated, dozens to even hundreds of genes all turn on at the same time because they all have the same transcription factor binding site on the DNA near the promoter. Okay? Okay, so now let's get into nuclear receptors, which are actually somewhat similar to SMADs. But this is a very well characterized class of receptors, and they are, even though they're called nuclear receptors, they are also a type of transcription factor, despite the name. So what they really are is kind of a single protein that has receptor qualities and transcription factor qualities. And they're called nuclear receptors because they are active in the nucleus. So let's look at this general structure of a nuclear receptor, and you'll see what I mean, that they're kind of a receptor and a transcription factor combined. So this is a diagram here of the protein sequence of kind of a generic nuclear receptor. And it's kind of stretched out from the N-terminal region of the protein all the way to the C-terminal region. And you can see that they have protein domains within them called A, B, C, D, E, F, okay? And I want to draw your attention to a couple of these particular domains because they're really important. One of them, E here, that's the ligand binding domain. So that is the portion of the protein that binds a ligand. Oftentimes these things bind hormones, incidentally enough. And so the ligand binding domain actually binds the ligand. And if you just design a drug that targets this nuclear receptor, that's where the drug will bind as well, okay? And oftentimes the drugs resemble endogenous ligands. Then there is a separate domain, the DNA binding domain, denoted by C here. That is the portion of the protein that actually associates with the transcriptional regulatory element that's right upstream from the promoter, okay? And so that allows this transcription factor to bind to the promoter. And interestingly enough, if you look at the 3D structure of these transcription factors, nuclear receptor transcription factors, the DNA binding domain has one structure. The ligand binding domain has another structure, and there's a little region called the hinge region, which is just a stretch of amino acids that connect these two functional units together into a single protein, okay? Now, interestingly enough, this thing typically does not act as a transcription factor until ligand is bound. So in the absence of ligand, this will not regulate transcription. So the DNA binding aspect of it requires ligand binding. Kind of makes sense. Okay. So a lot of times the ligands that bind these nuclear receptors are things such as steroids, thyroid hormones, in some cases lipids. They also associate with other transcription factors. They also have a propensity to dimerize before becoming active, and that we'll see how that is key in regulating where they are in the cell in a moment. Okay, and they do all sorts of things. These nuclear receptors can, some of the target genes include transporter genes, so genes that are involved in the transport of drugs into and out of cells. And a key to this also is that usually drug metabolism genes are regulated by nuclear receptor type transcription factors. So all of the CYP enzymes that metabolize drugs and are very important in pharmacology, they're regulated by nuclear receptors. So they're a very important type of transcription factor to understand because they're really important for drug metabolism. Okay, so these are transcription factors that are also receptors. They're kind of dual purpose. And here's what happens. I just showed you kind of the structure of a nuclear receptor, but here's how they work in a cell. And this is actually a subset called type 1 nuclear receptors. I'll tell you what the distinction is. But what happens is that in a cell that is not experiencing a ligand signal, these nuclear receptors are held in an inactive form, inactive. And what does that is a heat shock protein called HSP90. So HSP90s associate with the nuclear receptor and they hold them inactive, typically in the cytoplasm of the cell. Now, another thing I should mention about the ligands, for instance, the steroid hormone, these are the ligands that bind nuclear receptors must have the ability to get into the cytoplasm in order for them to encounter the nuclear receptor. So these are not receptors that are on the plasma membrane, rather they're kind of floating around in the cytoplasm. And certain ligands can actually penetrate the plasma membrane, and then they encounter the nuclear receptor in the cytoplasm. So the ligand, denoted by S here, binds the nuclear receptor, and when that happens, the HSP90s let go of the nuclear receptor, they dissociate from it, and now you have free nuclear receptor bound to a ligand. And typically what happens is that once that occurs, you need to have a dimer form between two ligand-bound nuclear receptors. So you'll have a dimer, and each of the monomers is associated with a ligand, in this case a steroid hormone. Once that dimerization occurs, this dimer is now competent to be imported into the nucleus, and there it will bind and associate with the transcriptional machinery, RNA polymerase, other factors, and then cause that gene to make pre-messenger RNA, which has introns in it. The introns get spliced out, all the exons get connected together. The mature mRNA is then exported from the nucleus to the cytoplasm, where it encounters ribosomes, and then you make proteins, okay? So that's how these nuclear receptors work. But you can see that the whole process is initiated by ligand binding, but the HSP90s need to be removed. Incidentally, this form of holding a receptor in an inactive form by associating with something else. In molecular terms, we call that sequestration. So these are sequestered, means they're held inactive, okay? So steroid hormones can actually readily diffuse across the plasma membrane, and then they encounter, this is kind of a summary, they encounter the nuclear receptor, and a lot of steroid hormone receptors are actually nuclear receptors. And then they typically dimerize and end up in the nucleus. This is a very simplified diagram here. I would study this one more. So there's a bit more complexity I want to mention. There are many types of nuclear receptors, and there are interactions between nuclear receptors, and they typically form dimers, but they can form homodimers and heterodimers, okay? So if the same kind of nuclear receptor dimerizes with itself, that's a homodimer, but if two different species of nuclear receptors associate with each other, that's a heterodimer. And so you get these mix and match combinations of nuclear receptors that then have different sets of target genes, okay? And so here is a nuclear receptor called RXR, that's the retinoid X receptor, and here's the estrogen receptor. So you can get like a heterodimer between estrogen receptor and retinoid X receptor, and that heterodimer is actually the one that can bind DNA. But it's even more complex with that. There are also co-activators and co-repressors. So co-repressors can associate with nuclear receptor dimers, but when a co-repressor molecule associates, this does not drive transcription. But if that co-repressor is replaced by a protein called a co-activator, now you have a fully functional nuclear receptor complex, and that will drive transcription. Okay, so co-activators and co-repressors are other proteins that actually regulate nuclear receptors once they're in the nucleus. And you can get this mix and match case. This is an example of a heterodimer, but there are also examples of homodimers that can actively drive transcription. So, what do these nuclear receptors regulate? Well, I already mentioned they regulate all the CYP genes, but another target of nuclear receptors are transporter proteins. And so there is a very famous protein called P-glycoprotein, which also goes by the name MDR, multi-drug resistance. And what happens with that is that when a drug gets into the cell, it can activate a heterodimer called PXR, SXR, and it drives the transcription of a gene called MDR1, which has a second name called ABCB1. That's an ATPase binding cassette protein. But basically, what this gene encodes is a protein called P-glycoprotein, PGP, and it will then embed in the membrane of a cell, and it forms a transporter pump, and it will actually pump drugs right back out of the cell, okay? Now, so it's kind of like a defense mechanism for the cell, and this usually happens in endothelial cells that line the blood. If your cells encounter a strange foreign substance that is not a part of your body, these cells have a way to pump that potentially toxic agent right back out of the cell. And that's why this system has evolved. But it's become a problem for chemotherapy because a lot of cancer drugs are actually toxic. And so cells get exposed to the cancer drug, they upregulate PGP, and that pumps the drug right back out of the cell. And so cancers experience mutations that drive cancer, But cancer cells also experience mutations that render them resistant to chemotherapy agents. And this is one such cancer resistance mechanism. But the whole thing is driven by nuclear receptors. Okay. So there are two different types of nuclear receptor classes, and they're based on cellular localization. Some nuclear receptors in their resting form are always present in the cytoplasm, and there's a second class that are always in the nucleus. And so you can classify them based on whether they're cytoplasmic or already nuclear. And you can also classify them whether they tend to form homodimers or heterodimers, okay? And so here's probably the most valuable distinction, and this has to do with whether they're in the cytoplasm or the nucleus. And so the ones that exist in a resting state in the cytoplasm are called type 1 nuclear receptors, and the ones that are obligately nuclear are type 2. So some examples of type 1 are androgen receptors, corticoid receptors, progesterone receptors, and estrogen receptors, okay? Whereas type 2, probably the most famous one is thyroid hormone receptors, but there are some vitamin receptors, you know, retinoic acid and vitamin D, another important class called PPARs, and bile acid receptors. So if you look at the evolutionary conservation of these, you can see that, you know, closely related receptors like estrogen receptor alpha versus beta are very similar, but there are others that are a bit different. And here's kind of the protein structure of all of these different nuclear receptors, a variety of them. But you can see that they all have a similar structure in the proteins. The N-terminal half of it contains the DNA binding domain, and the C-terminal half contains the ligand binding domain with a little hinge between them, okay? But the sequences of all of these are different, yet they still have these distinct domains within their protein structure. But again, the distinction between type 1 and type 2 is where they are in their inactive state. And so type 1 nuclear receptors are found in the cytoplasm when there's no ligand present, but when ligand binds, they can form dimers and then go into the nucleus, okay? So technically, type 1 can be either in the cytoplasm or the nucleus, but they'll only be in the nucleus if there's a ligand present, okay? Whereas type 2 are obligately nuclear. This is like the thyroid hormone receptor, for instance. And in this case, thyroid hormone, for instance, can go all the way through the plasma membrane and then be imported all the way into the nucleus where it first encounters the nuclear receptor, okay? So these are very special ligands. They have to be able to cross membranes very readily. All right, so type 1 nuclear receptors. They're also called steroid hormone receptors. As I mentioned, they form a complex with heat shock proteins when there's no ligand. So they're in the cytoplasm. They usually function as homodimers. And these homodimers form only in the presence of ligand. And then they go into the nucleus and regulate genes. And there's a lot of health relevance of these type 1 nuclear receptors. They're the targets of anabolic steroids, androgen receptors. They're also the targets of estrogen. And of course, that comes into play for estrogen replacement therapy in postmenopausal women, largely to avoid osteoporosis and the symptoms of menopause. And interestingly enough, these type 1 nuclear receptors, including the estrogen receptor, often drive cancers. So for instance, the estrogen receptor itself can drive breast cancer. Androgen receptors, so for instance, testosterone, can drive prostate cancer in men. These are also targets of anti-inflammatory drugs. So corticoid receptors are also type 1 nuclear receptors. So a lot of really important drug classes. And here are some examples of these. So it turns out that there are multiple different species of the estrogen receptor. There's not just one estrogen receptor. Estrogen receptors are a family. And so the concept of selectivity comes in again. And so here are some examples. Clomiphene and raloxifene. These are both antagonists of the receptor, so they're going to block the action of the receptor. And there's going to be competition, for instance, with plain old estrogen. So this is a fertility drug. And actually, males who are using anabolic steroids, that can cause stimulation of estrogen receptors, so they try to block that. Kind of an illicit use. But raloxifene is also an antagonist, and it can be used for the prevention of osteoporosis. And it's also used for prevention of breast cancer in high-risk patients that are predisposed for breast cancer. Here are some other famous selective estrogen receptor modulators, SERMs. A very famous one, tamoxifen, which is an antagonist. And it's a very common treatment for breast cancer and also for male breast cancer. Ormeloxifene, another antagonist. This acts as actually an oral contraceptive, and it's a common form of birth control used in India. So, interesting stuff. There are a lot of drugs. So if you look at these are antagonists, and here is the estrogen receptor, a type 1 nuclear receptor, and we're looking at the ligand binding region here. Here is the configuration when it binds estrogen, also called estradiol. And here's the configuration when it binds raloxifene. And you can see that the structure of the nuclear receptor is altered somewhat when it binds this antagonist raloxifene. And it renders this nuclear receptor incapable of acting as a transcription factor. So there's actually, when you treat people with raloxifene, there's a competition set up, competitive antagonism, between binding the drug, raloxifene, versus the endogenous ligand estrogen, okay? And of course, the affinities, the relative affinities for these two different molecules is going to determine which one outcompetes the other. So there are a lot of selectivity considerations that come into play with estrogen receptors. And so here are some of the different estrogen receptors, alpha-1, alpha-2, beta-1, beta-2, etc. And so here are a bunch of drugs, including raloxifene, and you can just see that this is a list of the pKa's. So this is estrogen itself, 9.4, but the pKa of raloxifene is 11.7. And remember, that is kind of the negative log. So the larger numbers mean tighter binding. And so you can see that estrogen receptor alpha has a higher affinity for raloxifene than estrogen. And that means that the raloxifene will bind the estrogen receptor more tightly, and it's going to outcompete regular estrogen, okay? And that's why it's a good antagonist, because of affinities. And it's also pretty selective too. You know, it's, well, it's somewhat selective. Look, I mean, here's raloxifene. It's binding HER alpha, but it also binds estrogen receptor alpha-1, not so much beta-1. So there are some selectivity things you can figure out here too. Interestingly enough, just estrogen itself has a similar affinity for ER alpha-1 versus ER alpha. And so selectivity is for endogenous ligands also. But anyway, what happens when you use a drug like this? Things like tamoxifen, raloxifene. What happens is these are drugs that can diffuse through the plasma membrane, and they activate the estrogen receptor, gets into the nucleus, and it has an unusual shape because it's binding a drug instead of the endogenous ligand. And then it prevents that estrogen receptor from activating gene expression. And so it turns out that the downstream genes from estrogen receptors in a breast cancer cell are genes that control the growth of breast cancer. So that growth of breast cancer is dependent upon estrogen signaling, okay? So if you block the estrogen signaling with tamoxifen or raloxifene, then the driver genes for breast cancer are silenced, they're turned off, and you can inhibit tumor growth that way. Unfortunately, a lot of breast cancers find a way to mutate their estrogen receptors, and then you get ER minus breast cancer, and the drugs don't work anymore because there's no estrogen receptor. So they find a way genetically to escape the control of the estrogen receptor, and that's a much more serious cancer. So anyway, how do they actually work? It turns out that estrogen receptors, once a dimer is made, they actually associate with regulatory elements of DNA. But in addition to things like, coactivators, co-repressors, they associate with chromatin remodeling proteins also. And so once the estrogen receptor is near a gene, chromatin remodeling proteins will loosen that chromatin, opens up the DNA, and now it becomes available for RNA polymerase to associate. So the actual mechanism whereby estrogen receptors actually work has to do with opening and relaxing chromatin. And there are a bunch of, there are a bunch of chromatin proteins, remodeling proteins here, HDAX, switch SNF, P300. You don't need to know all those, but I do want you to know that the actual mechanism at DNA for estrogen receptors, the reason they actually work is that they can change the local chromatin structure near a promoter. Because usually tight chromatin would block the access of RNA polymerase to a promoter. And so that's how these are physically working. So since these nuclear receptors, such as estrogen receptor, oftentimes drive genes that are involved in regulation of the cell cycle, when you treat with these drugs, it causes the cells to arrest their cell cycle. And this is using a procedure called flow cytometry, which I mentioned earlier. And this is actually a breast cancer cell line grown in a dish called MCF7. And here's what this breast cancer cell cycle profile usually looks like. You have a big peak in G1 phase and a smaller peak in G2. And in between, these are S phase, okay? So you remember the cell cycle G1, S, G2, M. This is a profile of a population of cells that are dividing, okay? So you have some cells in G1, some cells in S, some cells in the G2 phase. But when you treat those cells with an estrogen receptor antagonist, like raloxifene, for instance, they all pile up in G1. That means they can't progress to G2 or S or G2, and they actually can't do mitosis anymore, okay? And so this is a cell cycle arrest, and that's a very typical response of cells to being treated with an estrogen receptor antagonist, okay? Because the reason is the downstream genes that are regulated by that estrogen receptor transcription factor are genes involved in driving the cell cycle and cell proliferation. All right, so that's probably the most important stuff, but I want to briefly mention these type II nuclear receptors. And these are the ones that are obligately in the nucleus. So they're retained in the nucleus. These ones almost always function as heterodimers. And one of the favorite partners in the heterodimer is a nuclear receptor, a type II nuclear receptor called RXR. And these are typically, the way they're regulated is that the dimers in the nucleus are associated with co-repressors when there's no ligand present. But when ligand comes along, the co-repressor is replaced with a co-activator. Okay? And so here are some examples of these type II nuclear receptors. They're actually in the NR1 subfamily, but these are some of the most famous ones. The thyroid hormone receptor, retinoic acid receptors. There's another class called PPARs, that's peroxisome proliferator receptors, and vitamin D receptors, bile acid receptors. But we're going to look at thyroid hormone receptor kind of as a case study, and others. But here is levothyroxine, okay? So this is actually an agonist, and it looks a lot like thyroid hormone. So it's a thyroid hormone receptor agonist, and it's used to treat hypothyroidism and thyroid cancer. Here's gemfibrozil. This is a drug that binds to the type II nuclear receptor PPAR, and this one is used to treat dyslipidemias, diseases such as hyperlipidemia and hypertriglyceridemia. So you can already guess that, you know, the targets of these of thyroid hormone receptor and PPAR receptors are quite different. These PPAR tends to drive lipid metabolism genes. Couple of more. Tretinoin. This one is a retinoic acid receptor agonist, and that's called RXR. Useful for treating acne and certain leukemias. And here is vitamin D itself, which can be, you know, vitamins can be used as therapeutics. And the vitamin D receptor is a type II nuclear receptor. And it's used to treat rickets, which is just vitamin D deficiency. Also regulates low calcium. So vitamin D oftentimes gets added to milk because some of vitamin D can actually help release calcium stores in cells. Okay. So getting back to thyroid hormone receptors, the thyroid hormone itself looks like this, but it gets processed into different entities. So here is thyrionine. It can become iodinated to become thyroxine. So you can see iodine groups are added. And then it can be further modified, and that's called T4 hormone. It can be further changed into T3. So these are interesting hormones because they contain iodine atoms. And it turns out if you're near like a nuclear reactor that does a radioactive release, there's an accident, a radioactive iodine gets incorporated into these hormones, and then these thyroid hormones, guess where they accumulate? In the thyroid gland in your neck, and you can get a high dose of radiation from radioactive hormones. And so the solution for that is if you live near a nuclear reactor, sometimes the residents are given iodine tablets, and if you take a whole bunch of iodine that's not radioactive, it will displace the radioactive iodine and kind of wash it out, and you can survive this. But it's kind of, I think there were some iodine tablets near Millstone in Connecticut. People got those. It's near all nuclear reactors. Sometimes there's been a push to make iodine tablets available, just in case. But that's a different story. But in terms of physiology, here's a little bit more about the thyroid hormone system. And so actually, the pituitary gland, which is near the hypothalamus in the brain, actually, the pituitary produces a hormone called thyroid stimulating hormone, TSH. And TSH then stimulates the thyroid gland to start producing these hormone varieties called T4 and T3. And I just showed you those on the previous slide. So the thyroid gland is involved in iodinating those into active hormones. But once T4 and T3 are made in sufficient quantity, they actually inhibit thyroid stimulating hormone, and that's a self-regulating mechanism. So once there's plenty of T4 and T3 around, it will inhibit TSH so that excess T4 and T3 are not produced, and it reaches a stable equilibrium in most people. So that's kind of the biology. If you look at these type 2 nuclear receptors, again using thyroid hormone receptor as examples, once again there's actually at least 4 different thyroid hormone nuclear receptors, and they're all type 2, but they have a structure very similar to the type 1 nuclear receptors. They have a DNA binding domain near the N-terminus and a ligand binding domain near the C-terminus. But the difference is these are always in the nucleus, okay? But this is the region here that would bind the thyroid hormones, for instance. And here is how they work. The thyroid gland is kind of cranking out and producing T3 and T4. And in target cells, there are transporters that import T3 and T4 into the cell interior, okay? And then T3, which tends to be the most active of the hormones, can actually get all the way into the nucleus. And so now you're seeing how T3 actually gets into cells, T3 and T4. T4 can actually be transformed into T3 within cells. But there is a transporter in the cell membrane that allows T3 to get into the cytoplasm, and then it can actually diffuse right into the nucleus where it encounters thyroid hormone receptor, and it is typically complexed as a heterodimer with RXR, that's the retinoid X receptor, and once that ligand binding happens, a co-repressor is displaced, a co-activator associates, that's not shown here, but it's just as co-regulators, and then transcription can happen, okay? All right, so that's basically how that works. So, you know, if you look at drugs, here's T3 hormone, and here's a drug called DIMIT, and there are a bunch of them, but you can say this one has bromines on it instead of iodine, so an atom somewhat similar to iodine, and other than that, the structure is very similar. So that's a great hormone mimicking drug, and, you know, this just shows you a little bit that, you know, the same principles of a specific pocket, kind of a lock and key mechanism occurs for the binding of the hormone ligand to the nuclear receptor. So here is a thyroid hormone here, and it binds into a pocket on the thyroid hormone receptor. There are interactions with other amino acids of importance in the hormone receptor. And here it is again. If you had stereo glasses, you could see this in 3D, but it just binds in this ligand binding pocket. So even though this is a weird receptor, always in the nucleus, the same principles of ligand binding occur. There's kind of a lock and key fit mechanism for the ligand. Okay, so I think that concludes things for this part. And we've talked a lot about transcription factors. They're at the end of signaling pathways. On Friday, we're going to get into how do you actually figure out how many genes respond to a signaling pathway. What are the methods to do that? Okay? And we saw a few drugs that target nuclear receptors today also. All right, we'll see you guys on Friday, the last lecture day before Thanksgiving. So, very good.